Okay, in this video we're going to go through some revision notes for Biology 0610 Cambridge IGCSE for topic 10, Diseases and Immunity. So let's begin. Disease. Disease is a state of the body when it cannot cope with changes by normal homeostatic methods. So it's a state of the body where your body is not able to function properly. It's not able to cope with your normal behaviors of the body and normal processes of the body. Pathogen. Pathogen is a parasitic disease-causing organism. So pathogen is a parasitic organism that causes a disease. It causes your body to not cope with changes by normal homeostatic methods. Transmissible infectious disease is a disease by passing of pathogens from one host to another. So this is a disease that is caused by passing of pathogens from one host to a recipient. So disease is the state of the body when it cannot cope with normal homeostatic functions. Pathogen is the parasitic organism that causes this disease, and once this disease begins to spread from one host to another, it's known as a transmissible infectious disease. Transmissible infectious diseases can be transferred by two ways. The first one is by direct contact. Second one is by indirect contact. By direct contact, we mean that the pathogen is passed directly from a host to another by these methods: HIV, gonorrhea, hepatitis, or the most common one is transfer of body fluids as blood or semen. Sexual intercourse can transfer semen, blood donations can transfer blood, and kissing can transfer saliva. The second one is indirect contact. Indirect contact, pathogen leaves the host and is somehow carried to an uninfected individual. So any other way aside from the ways that are by direct contact. Here we have a methods of transmission table. So we have the method of transmission on the left side and examples of disease spread in this way. Droplets in air. Common is cold and influenza. Droplets in air can contain some sort of virus which can transfer cold or influenza to the recipient. Food or water, cholera, typhoid, and dysentery, usually if the food is infected or something. Touching contaminated surfaces, the most common one here is salmonella, usually in meat or athlete's foot. Insect bites is like malaria and fever. Examples of pathogens, the four common examples of pathogens present in our syllabus is bacteria, virus, fungi, and parasites. Now let's go through some body defenses. In our syllabus, we have 3 body defenses against such pathogens or diseases. The first one is WBCs, white blood cells. White blood cells prevent pathogens from reaching areas of the body in which they can replicate. So WBCs stop pathogens from going to areas where they can reproduce themselves. There are 2 types of WBCs in our body: phagocytes and lymphocytes. Phagocytes perform phagocytosis by engulfing and digesting enzymes. They capture the enzymes, engulf them, and produce digestive enzymes to destroy them. While lymphocytes, on the other hand, produce antibodies to perform agglutination. The function of lymphocytes is antibody production, which causes agglutination. Agglutination is the clumping of pathogenic cells together to restrict their movement and release chemicals other cells to be destroyed. So agglutination is when a bunch of pathogenic cells clump together. This restricts their movement. They're not able to move very easily. Due to this, a chemical is released, and this chemical signals phagocytes to come, engulf the pathogenic cells, produce digestive enzymes, and destroy them. The second body defense is chemical barriers. Chemical barriers are substances produced by body cells to trap and kill pathogens before they cause disease. So they are substances produced by our body cells, and they trap and kill the pathogens before they can even cause disease in our body. We have 3 chemical barriers. Well, we have many, but in our syllabus, 3: mucus, stomach acid, and lysosomes. Mucus is to trap pathogens and remove from the body by coughing or blowing nose. Usually when you're coughing or you're blowing your nose, you feel some sort of mucus. This is a sort of chemical barrier which kills the pathogen before it causes a disease. The second one is stomach acid. Our stomach contains hydrochloric acid, HCl, to kill any pathogens caught in the mucus because it provides acid-low pH. Lysosome is the third one. Lysosome is present in eyes to destroy bacterial cell walls in tears. The third defense in our body is mechanical barriers. Mechanical barriers are structures that make it hard for pathogens to get past them into the body. So they make it hard for pathogens to even enter the body. We have two mechanical barriers, skin and nose hair. Skin covers almost all parts of our body, and it prevents infection from pathogens, and it can also heal itself. Nose hair causes difficulty for pathogens to enter and reach the lungs because of the high concentration and they're clumped together. So this way we have finished the types of defenses in our body. Now let's see how we can control the spread of disease. By controlling the spread of disease, we mean we have to stop the pathogen from spreading by good hygiene, effective sanitation, waste disposal, and sewage treatment. So let's have a look at this table here. In this table, we have the measure to prevent the spread and how it works. The first one is hygienic food preparation. Hygienic food preparation consists of the following points. Number one, keep food cold so bacteria and fungi reproduce slowly, because at low temperatures, the reproduction rate of bacteria and fungi is reduced. Prepare food hygienically to avoid contamination from pathogens by washing hands with soap. Cook food long enough at high temperatures to kill bacteria and fungi. So before keeping the food cold, we cook it at high temperatures to kill as much of bacteria and fungi as we can. After that, we keep it at a cold temperature to reduce its reproduction rate. Cover the food to prevent flies on it before eating and other bacteria and dust particles. Use separate chopping boards and utensils for cutting uncooked meat to prevent bacteria from spreading. Wash hands after and before using the bathroom. The second one is personal hygiene. Washing hands with soap before removes pathogens from the skin. Use tissues to catch sneezes and coughs. Dispose used tissues as soon as possible and wash hands after and before using the bathroom. The third one is waste disposal. Waste food should act as vectors for transmissible diseases, and they should be disposed in a sealed container. Rubbish bins should be covered from the top. Don't leave them open from the top. Sanitation is the last one. Homes should have plumbing and drains to remove faces and waste. Sewage should be treated to remove solid waste. Okay, so we have finished everything about diseases. Now let's jump to immunity. Immunity is the defense to a pathogen. Pathogen is, just to recall, a disease-causing organism, and immunity is what defends against this disease-causing organism. We have two types of immunity, active immunity and passive immunity. This is an intro summary table before we jump to them in detail. So active immunity, the production of antibodies is by the body, while passive immunity is ready-made. The time before antibodies appear in the blood in active immunity is 1 to 2 weeks, so it takes a lot of time in active immunity, while passive immunity is immediate response because it's ready-made. Active immunity has presence of many cells, yes, because it's long-term immunity. Passive immunity does not have presence of many cells because it's short-term immunity. So types of active and passive immunities. So these active and passive immunity are divided into further two more groups, natural and artificial. Active can be natural by exposure to a pathogen. Active can be artificial by vaccination. We're going to learn them in detail in the upcoming slides. Passive can be natural, given by antibodies from the mother via placenta or breastfeeding. It can be artificial, manufactured, and injected. This is a graph not required for A-levels, but just to understand the primary and secondary response to a pathogen. So on the graph, as we see, the primary response to a pathogen after the first pathogen injection, we have a low concentration. But after the second pathogen injection, memory cells are produced and there's a high concentration. To understand this, we need to understand memory cells, which we're going to do that in the upcoming slides. Here's another intro summary to types of immunity. This one is a bit more detailed. So immunity may be active or passive. Active is when the individual makes his or her own antibodies. The body makes its own antibodies in active immunity. Passive is given ready-made antibodies. Somebody else gives you antibodies in passive, while active you already have them. Active immunity is long-term because memory cells are present. Memory cells are cells that retain the ability to produce antibodies. They remember the instructions, so if the infection is encountered again, they can fight against the infection. Passive immunity has no memory cells. This is why passive immunity is short-term immunity. Active can be natural or artificial. Natural is if pathogen infects the individual. What happens in natural is that a pathogen will infect the individual. The individual will get sick for some time because the lymphocytes are preparing antibodies. After some time, when the lymphocytes have prepared antibodies, the lymphocytes will fight against the pathogen and they'll cause agglutination, destroy the pathogen. After the process is done, the instructions for this pathogen is going to be stored in memory cells for further infection. During this time, the individual may get sick. Artificial is by vaccination. Vaccination does not cause any disease, it's just a dead or altered form of a pathogen injected into your body, so memory cells can retain its instructions for further infection. Passive natural is when the mother gives you antibodies when you're in the placenta or via breast milk. Artificial is by injection. Immunity is the body's use of antibodies to combat invasion by pathogens. Vaccination or vaccine is a chemical or cell which provokes the body's immune system to make antibodies. So let's go into further detail about active immunity. Active immunity is making antibodies and developing memory cells by the body for future response to infection. So the antibodies are made by the body, they develop memory cells, and they are long-term because they have future response for infection. Active immunity can be by two ways. The first one is the body gets infected by a pathogen and the lymphocytes makes antibodies specific to that pathogen. So during active immunity, the pathogen and the antibodies are like the enzyme and substrate complex. The enzyme and substrate are complementary in shape to each other, so are the antibodies and pathogens. Lymphocytes will always produce antibodies complementary in shape to that pathogen. The second way is vaccination. Active immunity is slow acting and provides long lasting immunity because presence of memory cells. Some key definitions: antigen is a protein molecule found projecting on the surface of the cell membrane. This one is to be destroyed by the antibody. Antibody is a protein made by lymphocytes that's complementary in shape to an antigen, and when attached, it comes together and signals the cell to be destroyed. This is known as agglutination. Antitoxin is a protein that neutralizes the toxins produced by bacteria. Now let's have a look at how active immunity works. So during active immunity, lymphocytes read the antigens on the surface cells and recognize any foreign ones. This means we have a cell and there are antigens on the surface of the cell. If there are any foreign antigens that can cause infection or disease, a lymphocyte will read them and identify them. As soon as a lymphocyte identifies any foreign antigen, it's going to make antibodies complementary in shape to that antigen on the surface of the pathogenic cell. These antibodies will then go and they will attach to the antigens and they will cause agglutination. They will come together and restrict their movement, and then they will signal phagocytes to release a chemical and to be destroyed. So again, lymphocytes will read the antigens on the surface, recognize any foreign ones, make antibodies and complementary in shape to the antigen. They will attach, cause agglutination. They will signal phagocytes. Phagocytes will come and they will destroy the antigens. Now, during active immunity, the initial response for a lymphocyte encountering the pathogen for the first time to make specific antibodies will take a few days because it's the first time. During this time, the individual may get sick, as we said before. The lymphocytes that are made for the antibodies for the specific antigen will then make memory cells, cells that retain the instructions for the specific antibodies for that pathogen. So this way, in case of reinfection by the same type of pathogen, the antibodies will regenerate quickly and they will destroy the pathogens before they multiply to cause illness. This is how people become immune. They encounter a pathogen, develop memory cells, they become sick, then develop memory cells. After that, the infection will not encounter or hurt them again because they're immune. Why? Because the instructions for this antibody and antigen is restored in the memory cells. antibodies are always Y-shaped. So here let's look at the diagram of how active immunity is working. Over here we have a lymphocyte, and this is a pathogen. So this is the antigen on the pathogen, and we said Y-shaped are always antibodies. The antibodies in complementary in shape to the antigen is going to attach to the antigen, and it's going to come together and cause agglutination. After that, it's going to signal the phagocyte to come and engulf the antigens and cause agglutination, destroy them, and then memory cells restore the instructions. Our second method of transfer in active immunity is vaccine or vaccination. A vaccine is a protection against a specific disease to boost the body's infection defense against it. So vaccinations are only for specific diseases. And how do they work? So vaccinations or a vaccine inputs dead or altered form of the disease-causing pathogen that contains specific antigens introduced into the body. So it's going to put a dead form of the disease-causing pathogen into the body. In this state, the pathogen cannot cause any disease because it's dead, but it will provoke an immune response because lymphocytes will identify the antigens, and lymphocytes will make memory cells to restore the instructions. So in case in future, if a live pathogen of this same species is encountered again, memory cells can quickly respond and destroy it. The level of protection against specific disease depends on the proportion of people vaccinated, and since vaccines use memory cells, it's long-lasting immunity. Any immunity that is using memory cells is long-lasting immunity. Herd immunity. So I'm sure we've heard this term herd immunity a lot in COVID. Herd immunity is when a large percentage of the population is vaccinated, resulting in a few places for pathogens to breed and provide protection for the entire population. So the main concept of herd immunity is just to keep the population of vaccinated people very high, because this will reduce the rate of people who are being infected and reduce the rate of pathogens reproduction. Herd immunity prevents epidemics and pandemics because, of course, when a large percent of the population is vaccinated, fewer people are going to be infected and fewer diseases are going to spread. If the number of vaccinated people drops in the population, there's a mass infection risk, and number of people in the population could increase of death. Children are regularly vaccinated to keep the vaccination population at a high level. This is why children up to 6 years old are regularly vaccinated. Smallpox is an example of a disease that was successfully eradicated in the 1980s after a vaccination program run by World Health Organization, WHO. This is a diagram of how the vaccination is working. So we have a human, we're injecting dead antigens inside this human or dead pathogens. Lymphocytes will recognize this, but it will provoke an immune response. During this time, the individual will not get sick, but memory cells will be prepared in the human's body for future response to a live pathogen. This is a herd immunity diagram. So as we can see, let's look at the key first. The key says blue is not vaccinated, but still healthy. Green is vaccinated and healthy. And red is not vaccinated, sick and contagious. So going back up, in the first part, we have a lot of not vaccinated but still healthy people, and then we have sick people. Since no one is vaccinated, the contagious disease spreads throughout the whole population and number of sick people increases. In the second one, some of the people are vaccinated but not all of them, and some of the people are also sick, but the number of sick people that has increased is not as great as the first one. In the second one, most of the population is vaccinated, only 2 are sick, and 2 remain sick. And that is all for herd immunity. Now coming back to the major immunities, passive immunity. Passive immunity is not so detailed, it's a short one. Passive immunity is just a fast-acting and short-term defense against pathogen when antibodies are received from another individual. So passive immunity does not have any memory cells, it's very fast and legit. A person just receives antibodies from another individual, is fast-acting and short-term. It happens either 2 ways: mom to infant via breast milk, mom to infant via placenta. It does not make antibodies or memory cells, and it helps the young to grow older because it's usually given to infants. That was all for active and passive immunity. The last part in this chapter is chlorea. Chlorea is an infectious disease that is caused by a bacteria known as Vibrio cholerae. Vibrio cholerae is a bacteria that causes diarrhea. Diarrhea is loss of watery feces from the anus. So cholera is just diarrhea. Diarrhea caused by a specific bacteria known as Vibrio cholerae. Diarrhea is when water feces is lost from the anus. Severe diarrhea can cause a lot of water and ions lost from the body, and this may lead to death. The treatment for cholera is oral rehydration therapy. Oral rehydration therapy is a drink with dissolved salt and sugar. So let's see how Vibrio cholerae causes diarrhea. First, there is an ingestion of infected water or food into the small intestine. So the human ingests infected water or food. This infected water or food contains bacteria. This bacteria attaches to the wall of the small intestine and it will produce a toxin. So you ingest infected food which contains bacteria. The bacteria attaches to the small intestine, produces a toxin. This toxin stimulates cell linings of the intestine to release chloride ions. So simply, the toxin releases chloride ions into the lumen of the intestine. These chloride ions will accumulate in the lumen and lower its water potential. Of course, when we hear the term water potential, we know we're referring to osmosis, because since the water potential is lowered, water begins to move out of the cells into the large intestine via osmosis. So again, the bacteria produced a toxin. This toxin released chloride ions. The chloride ions reduced the water potential, and since the water potential was lowered, water started to move from osmosis from the small intestine into the large intestine. From the large intestine, a huge amount of water is lost as watery feces, and the blood will contain little amount of water and ions. And that is it for the process of chlorea. Let's just refer to the diagram here to understand it. So this is the large intestine, this is the small intestine, this is a cross-section of the small intestine, this is the wall lining of the small intestine. What's happening is that bacteria has entered the small intestine and it affects the lining of the small intestine. Due to this, water potential is being lowered because water is being released outside and a bunch of chloride ions are being released outside. This is the toxin that is attached to the lining, which releases these chloride ions, and then via osmosis, water is being excreted out of the small intestine into the large intestine and will be released as watery feces to produce diarrhea.